Hello, my name is Michelle, and I'm going to be reviewing some frog embryology with you today. So, this is an important topic to actually tack on as we go through human embryology. Just because it is fairly high yield for the DAT, it's pretty common for people to encounter a question or even a couple questions about frog embryology. So, I would recommend reviewing this once you've already gone through human embryology and you feel like you have a pretty strong foundation with the processes as well as some of the most important structures. Okay. So, after fertilization, as we remember from human embryology, we're going to go into cleavage. And as we may have mentioned before, there's a couple different types of cleavage. The first main category that we talk about is holoblastic versus meriblastic cleavage. And as you can kind of tell from the name, holoblastic, it has the word whole in it. That means that the zygote that is now fertilized is going to undergo cleavage in a whole and a complete way. So basically, that initial cell is going to divide into two. It's going to be an even process. That two is going to go into four, and so on, as you can see at the top image here. On the other hand, we have meriblastic cleavage, which physically is different just because it's uneven. Now, we'll start to go into the reason why this happens. And the primary reason for that is because with holoblastic cleavage, this happens in organisms that tend to have only a little bit to a moderate amount of yolk. So that's not preventing the even cleavage. On the other hand, with organisms that do tend to have a lot of yolk, it's more common for meriblastic cleavage to happen. And this is because when a cell is trying to divide, the yolk kind of blocks that process. So it ends up being uneven, as you can see here. And a lot of the cells are going to look a little bit different. Now, the most notable thing about this is that even though frogs do have a lot of yolk as an embryo, they actually are surprisingly undergoing holoblastic cleavage. So holoblastic cleavage, just like humans, just like any other organisms that tend to have less yolk during the process of embryogenesis. So that's the most important takeaway from this slide. All right, this is just to show you an image, not a cartoon, a real image of what a frog egg looks like undergoing cleavage. So this is a scanning electron microscope image. As you can see, it's pretty even going through these different slides. It looks even all around, and it's complete cleavage. Okay, so now that we've covered holoblastic cleavage, the fact that frogs undergo holoblastic cleavage, there's another detail that's really important, and this is a way in which human embryos are different than frog embryos, and this is polarity. So if you look at the right-hand side, this is sort of a sliced image of a blastocyst of a frog blastocyst, basically. So the top half is going to be what we call the animal pole. That's the blue part here. As you can see, the cells are a little bit denser compared to the vegetal pole, which is on the bottom. So the main difference between the animal pole and the vegetal pole is that there's a lot of active division that's happening at the animal pole. So as you can see, the cells are denser. They look a little bit smaller and closer together. That's because it's undergoing rapid division compared to the vegetal pole, in which the division is going to be a little bit slower, and this is going to be, eventually where the yolk is. So that part of the cell is going to develop more into the structures that are giving nutrients to the cell. So it's not really as important, or to the organism rather, so it's not really as important that we're dividing rapidly here, whereas on the animal pole, this is the active side, that's what's actually developing into the organism itself. So physically, they look different on this slide. If you were to look at it under, a microscope, what you would see is that the animal pole is going to be darker, right, because it's denser, so it'll appear sort of a black color. On the other hand, the vegetal pole is going to be a lighter color. Now, you may be wondering, okay, how am I going to distinguish between these two different names? And what you can think about is the word vegetal, it sounds kind of like, the word vegetable, like vegetative. So when we think of the word vegetative, that's something that's kind of lazy, isn't really doing a whole lot, mostly just lying around when you're in a vegetative state. So that can remind you of the vegetal pole, which actually is not undergoing a lot of division. And that's, I think, for me, the easiest way to understand it. Now that we've distinguished the different poles, it's important to note on the left-hand side that typically organisms undergoing holoblastic cleavage do not have this polarity. So humans don't have this polarity, sea urchins don't have it. So if you look at the top, that's where the sea urchin cleavage is going. So as you can see, it's undergoing holoblastic cleavage, and there are cells that are divided evenly. Whereas underneath we have the frog cleavage. So as you can see at the top, there's the animal pole. At the bottom, there's the vegetable, and the division is not quite even. As we move towards the right, you can see that the cells at the animal pole are a little bit smaller, and at the vegetable pole, they're a little bit bigger. Okay, so now that we've covered that polarity, we're going to now move into a new important structure that you definitely should know for the DAT. This is the gray crescent. So the gray crescent, I'm just going to orient you where we are looking at this organism just so you can see how this forms. So if we're looking at this kind of section of blastocyst, basically the top half of it, as you can see, it's gray, but it actually should be black. So that's going to be the animal pole. The bottom half that's white, that's going to be the vegetal pole. So the sperm is going to enter anywhere in the top half where the animal pole is. At the opposite end of that, we're going to have the formation of the gray crescent. Now, the reason that the gray crescent forms is actually because it's a mixture of the black appearance of the animal pole and the white appearance of the vegetal pole. And this is because after the sperm enters the egg, basically the cell is going to rotate a little bit so that there's a little bit of mixing. As you can see in this image, it rotates about 30 degrees, and that's where the gray crescent forms. Okay, so the last important structure that we're going to talk about is the dorsal lip. And just to orient you kind of where we are, this is once we've gone through fertilization, we've gone through holoblastic cleavage, and we're beginning to go through blastulation. So basically, we're at the stage of a blastocyst. And if you recall from human embryology, there's a very important structure in the blastocyst that determines where gastrulation, or formation of the 3 germ layers, is going to occur. And this is really important because it helps orient the rest of basically like where the whole embryo is going to form. So we actually don't have the primitive streak in frog embryology. What we have is called the blastopore. So you can see that at the bottom. That's going to develop as we go through gastrulation. And the part of that we need to remember for the DAT is the dorsal lip. That is the site that determines where gastrulation occurs.